Thank you for joining me for an additional video update and installment talking about biochemistry, or at least an introduction thereto. In this video, I'm going to teach you guys about amino acids, proteins, and carbohydrates. Are you ready? Let's begin. So as I mentioned in our last video, there are four classes of molecules needed to sustain life: proteins, carbohydrates, lipids, and nucleic acids, and a picture of Dr. Evil just for fun. I'm now going to introduce you to, in this video, amino acids, and by extension proteins and carbohydrates. So much like a house is made of bricks, concrete, wood, and so forth, proteins are made of 20 building blocks called amino acids. Amino acids then, as we can see in this cute little diagram, are the building blocks needed to be strung together to make a protein. Proteins are found in nearly all living tissues. One of protein's most important roles is to serve as biological catalysts called enzymes. And yes, life could not exist without them. So, as I mentioned before, amino acids are the building blocks used to make proteins. Amino acids are simple molecules that look kind of like the one shown right here. They're called amino acids because they have both a carboxylic acid part, which generically speaking is a molecule that looks like this. So you can see the carboxylic acid part right here. And they also have an amine part. This is what an amine looks like, so you can sort of see this amine dangling off of here. These are two functional groups, by the way, that we learned about in our organic chemistry section. The letter R here in this structure is a generic letter that means any chain of carbons or hydrogens. Now, in the case of specific amino acids for life, there are only certain R groups that I'll show you momentarily. One thing I want to point out is this. The carbon shown right here is called the alpha carbon, the one that's sort of wedged between the amine group and the carboxylic acid group. So the only real difference then between one amino acid and another is the identity of the R group attached to its central carbon, called its alpha carbon. You should notice then that if the alpha carbon has four different groups attached to it, which will depend on the identity of its R group, then the alpha carbon will be a stereocenter or chiral center that we talked about in an earlier lecture. Now, just so you know, there are 20 amino acids that are used as building blocks for all proteins found in all living organisms. These 20 amino acids, which I personally call the 20 amino acids of life, are shown here in this table. You'll notice that all of the individual R groups, these side chains attached to the alpha carbons in the amino acids, are highlighted in blue. So those are really the only structural difference separating one amino acid from the next. Now, please note that glycine, which is this amino acid right here, is the only amino acid that does not possess a stereocenter. The reason is because its R group is actually a hydrogen. So that means that the alpha carbon in the center, this one right here, is attached to two hydrogens. Because it's attached to two hydrogens, that alpha carbon is not attached to four different things, and is therefore not a stereocenter. We say then that glycine is achiral, and it is the only achiral or non-stereocenter possessing amino acid of all of the 20 amino acids of life. Now, when the amine group from one amino acid replaces the OH group on the carboxylic end of another amino acid, here's what happens. So I've got my amine group on one amino acid staring at the OH group of another. If this condenses so that the nitrogen takes the place of the OH and kicks off the OH as water, we get this product right here. Now, the bond that I bracketed right here is called an amide bond, and the reason is because this type of functional group happens to be called an amide. Now, I want you to keep that in mind momentarily as I go on to the next slide. Just so you know, single bonds, unlike double and triple bonds, can rotate freely in three-dimensional space. Hence, if we rotate around the bond shown right here, we can see that this molecule right here could be redrawn as the molecule shown here to the right. These two molecules are exactly the same. You'll notice that, once again, if I just rotate around this, that will end up poking this oxygen down, and this group, which is pointing three-dimensionally away from us, will end up poking three-dimensionally towards us after I do the rotation. Hopefully you're okay with that. Anyway, at this point, I want you to keep this in mind as we go on to the next slide. So what happens if multiple amino acids get together? Well, if multiple amino acids get together and do the same thing, each amine group from one amino acid can condense onto and take the place of the OH group in the carboxylic acid of the amino acid next door. If that happens again and again and again, you get a big long chain that looks like this. Now I've placed little red brackets here to point out the repeating amide bond units, which are called peptide bonds. Now, a chain or polymer of amino acids like this one is called a peptide. Now, in fact, as it turns out, a chain of 2 amino acids is called a dipeptide, of 3 amino acids is called a tripeptide, of 3 to 10 amino acids is called an oligopeptide, and of up to 50 amino acids is called a polypeptide. A chain of 50 or more amino acids altogether is called a protein. So in essence, a protein is a polymer of amino acids. The thing that causes one protein to be different from another are the identities, once again, of the R groups, called side chains, from one group to the next. This is determined, of course, by which of the 20 amino acids of life is found at each position in a particular protein's chain, as well as the total length of the chain itself. Do you like that? I hope so, because that's all I have to teach you about amino acids and proteins. Now I'm going to teach you about carbohydrates. So the term carbohydrate is a fancy way of saying sugar. Complex carbohydrates, or polysaccharides, are made up of multiple smaller sugar molecules bonded together. Simple carbohydrates, or monosaccharides, are just one sugar molecules, like glucose and fructose, whose structures are shown here. Sucrose, shown right here, which is the structure for common table sugar, is a disaccharide made by bonding glucose and fructose together. Thus, you can see that if you take sucrose, table sugar, and you stir it around in aqueous acid, you can actually get it to separate apart into individual glucose and fructose molecules. Monosaccharides, or simple carbohydrates, can exist in 2 forms: an open chain form, drawn right here, and a cyclic or cyclic form, or suclic or cyclic, depending on how you pronounce it. Some of those might have been absolutely ridiculous pronunciations, but, you know, I like the ridiculous. This is the cyclic form of glucose, this is the open chain form of glucose. And these are actually the same molecule. Now you might be wondering then, how are these 2 molecules the same molecule? They look totally different. I'll show you. As it turns out, glucose can actually interconvert between these 2 forms, the open chain form and the cyclic form, by doing this. If you can imagine this green OH right here, if you actually redraw this open chain form, to kind of grab that OH with your hand and bend it around, you can sort of see that it looks like this. And if this green OH condenses onto this carbon, forming a bond between this oxygen and this carbon, and then this H gets removed, the electrons pump up onto this oxygen, and then that oxygen gets protonated to form an OH. This is how this open chain form is converted into the cyclic form. And if this is sort of confusing to you, I want you to pause the video and look at it really closely until it makes sense. So in reality, glucose is actually interconverting between these 2 forms constantly. The cyclic form is the more stable one, so it tends to exist in this form in a larger amount, percentage-wise, than the open chain form, in which it only exists transitorily. Alright, now as I indicated a few slides ago, simple carbohydrates like glucose and fructose can be bonded together to form disaccharides, such as sucrose. Similarly, when organisms bond many simple carbohydrates like glucose together, they can form huge polymers like cellulose, which is the major material used by plants to make leaves, stems, roots, bark, flowers, and pretty much all of physical plant matter, or the majority of it. So in essence, if I take about 3,000 glucose molecules and I bond them all together in a very specific way, I can get this repeating polymer of basically this unit shown over and over and over and over again in both directions. And that polymer is cellulose. Once again, cellulose is the major constituent in the vast majority or in most of plant matter and gives plants its structure. Pretty cool, huh? So that takes us to the end of this video. Please stay tuned to the next one, in which I'll teach you about lipids, also known more popularly as fats. Until next time then, my wonderful students, have an enjoyable rest of your day.